So just to recap, depolarization carried by sodium, depolarization carried by a different one, calcium. Sodium channels inactivate, but instead of continuing to repolarize, it's delayed. There's an influx of calcium surges inside the cell, and it keeps the cell positive, and it delays its repolarization. Okay, so that's important. And the importance of that I'll emphasize more later, but for now, just kind of like write that down. So I'm kind of trying to color code because there are differences between these two cells. Now for the third one, the potassium current repolarizes both cells. So to repolarize to a negative potential. So green is for potassium. All right, so the fourth thing is the pacemaker current. They're called funny currents. Now, some of that current is carried by sodium. So that's that pacemaker current where it's a slow rise to threshold. And then you get your action potential. And then when you get to a negative potential again, it doesn't stay there. It's unstable. And it just wants to rise the threshold again to give you another action potential. And again, the depolarization is carried by calcium. And the repolarization is carried by green, potassium. So I drew a second action potential there. So for here, what I had said was there is a resting potential for this action potential, and it's carried primarily by potassium current. So I'll use green for that. And so this is what I was trying to draw in your notes. You already drew these, right? Just make the connection in your notes. Remember how I drew this before? And I drew that before in your notes. I just want you to know what current carries what. Okay, and let's review all the potentials. This is the action potential, right? Action potential for both of those. What kind of potential is that? The yellow line. Pacemaker potential. Yeah, that's what we said before. Where's the pacemaker potential here? There isn't one. Where's the threshold potential? There isn't one. You go straight from rest to action potential. So we call that negative 90 resting potential, I guess, if you want to call it something. But the main thing to teach is the action potential. Okay. And you're supposed to know what current carries what. So let's kind of move away from this. And let's do a deeper dive into the contractile cells. Those cells, remember, they can generate tension. So look at the top graph. Remember when I drew this before, I drew a tension curve? Because we want students always to remember that these are the cells that create tension. These do not. And so, yeah, let me erase the board. But we're going to look at this top graph here, and we're going to relate it to this bottom graph here. Maybe I can leave that side, but I want to erase this. We'll come back to autorhythmic cells. So why did I use yellow, red, and green? match these colors for this slide. If you didn't use the same colors, it doesn't matter. I just used it so it could not confuse colors. Now I put in that 0, 1, 2, 3 because I want you to know what kind of happens at each of those places. So where I put the 0, that's rest. And just kind of look at both graphs. The top graph is millivolts. The bottom graph is the whole permeability thing. What is the cell permeable to? So let me take you through how you read it. At rest, the millivoltage is -90. It's about -90. So what we want students to see with this bottom graph is, how is it -90? What is the cell permeable to? And if you look at our three currents, the one for sodium, calcium, and potassium, the cell is most permeable to, what's the highest value? Not sodium. Look where the 0 is. See, I see red at the bottom, then I see a little bit of yellow, then I see green. What is green again? So where the 0 is, the cell is most permeable to potassium. Now, if you remember, potassium, the gradient is always set up where potassium leaves the cell. So if you have potassium leaving the cell, that makes the inside of the cell negative with respect to the outside, for the membrane potential. So, I don't know, does that make sense to you? Positive leaving cell, this membrane potential is negative. So I'm trying to teach you why the membrane potential is -90. Don't just memorize it's -90. Memorize it because the cell is more permeable to potassium in the condition of rest. Okay, that's the key. Now, look where I put number 1. I put number 1 right by the depolarization. What causes depolarization? You just wrote it here. Sodium. The sodium, where I put the number 1, is the highest. Okay? I mean, potassium and calcium don't even come close. You get this powerful sodium influx. That's making the membrane potential positive with respect to the outside. I mean, the permeability to sodium is so high, it's off the chart. Okay? So that's basically what I want you to know there. You have voltage gated sodium channels. You remember studying the voltage gated channels in 4.3? That was really important, actually. These voltage gated channels for sodium and potassium, they're one of the most studied if you go into neurophys. So the idea is, in your head, picture a sodium channel, a couple of gates, and maybe there's a gate where, I'll draw this gate like this. Draw a couple of gates. Well, anyways, this gate here, the way I explain voltage gated, you have to understand as a student what opens a channel so the cell is permeable to it. There's a gate on it, okay? And you can open or close the gate in different ways. If there's a change in voltage, that'll open or close the gate. So imagine there's a little positive charge on that part of the gate when it's closed. So imagine there's some gap junctions here. Now don't imagine, this is how it really works. but you have to imagine because you can't see it under the microscope. This is how the signal spreads through the intercalated discs. You go straight from rest to depolarization. Because when positive charge gets into the muscle cell, from like the SA node spreading it everywhere, the positive charges go right through and they depolarize the cell. When those positive charges get close to that positive charge in the gate, positive repels each other. So that swings the gate this way. So that way the gate opens. Okay, so that's what you have to understand. And you get this huge sodium influx, and that causes the depolarization. Okay, that's all I want you to understand. But I'm going to move on now. That's depolarization. Now, any questions on depolarization? How you open the gate? So this is a V-gated sodium channel. If you take an upper division neurophys course at a university, they'll get way more detail on that. You just have to basically understand that these things are gated, and when the gate is open, the cell's permeability to that ion changes. So these sodium channels, they open and close really fast. These other channels, they're also voltage gated. Their kinetics are just slower. They open and close slower. The sodium channels open and close before the other ones even have a chance to open or close. Okay, that's kind of what we see here. I always give the analogy of like a 100-meter race, right? And I always give the example, if I race like Usain Bolt, I would lose the race. But we both start at the same time, but we both finish at different times. Okay, that's like the sodium channel. They just open and close super fast. And that's what I want you to understand. Now, as fast as they open, they inactivate. As soon as they open, this gate is the inactivation gate. Inactivation gate. It's gonna plug it up. I can't illustrate everything, but that causes that little downtick. Okay, that's why you start to repolarize, because you plug it up super fast. So that's why you see this rapid drop in sodium. That's sodium channel inactivation. So I'm going to write here as a part of number 1. Inactivate fast. However, you don't continue to repolarize. That repolarization is delayed by calcium channels. So I'm going to move on to where number 2 is. I put the number 2 right in the middle of this entire area right here. I call that the plateau, the flat part of the action potential trace. And it's caused primarily by calcium. So there's a lot of things that happen during number 2. The plateau is a delay in repolarization. What's going to happen is voltage-gated channels, voltage-gated calcium channels are going to open. You're going to get this influx of calcium. It causes this, I believe your book might call it the calcium surge. Well, let's just call it that. So imagine you have calcium channels in the cell membrane. I'll just draw them like this. They're in the cell membrane. And I don't know if you remember the T tubule. The T-tubule is a structure that allows the action potential to spread across the surface, but then deep down within the structure of a cell. Anyways, in there you have these other structures called the Sarcoplasmic Reticulum. And in muscle cells, they contain lots of calcium ions. Where I'm going with this is, you want to liberate this calcium. You need enough calcium for the cross-bridge cycling so the muscle can contract. Okay, and well, how you do that is this calcium surge, or extracellular calcium, comes into the cell. You know, that changes the membrane potential. It keeps it depolarized for a while. You're increasing, this is a very long action potential. Instead of being 10 milliseconds, it's like 250 milliseconds. It's very long. Okay, this whole, it's a long action potential. The thing about it is, this intracellular calcium isn't enough. Let's remember that you have the contractile proteins, you know, actin and myosin, the thick and thin filaments. There's myosin, there's another myosin. You have the M line, you have the actin, the overlap, the details of the sarcomere. And anyways, you need enough calcium to get the cross-bridge cycling going. This intracellular calcium that comes in is not enough. You need to kind of trip this thing open. Now there are different names, but there's a voltage, there's a calcium-sensitive channel that opens this sarcoplasmic reticulum. So let me draw like a little channel in here. How about I just draw like a, I don't know, something to symbolize some kind of gate that can open. It's a calcium sensitive gate. Different books call it different things. Sometimes it's called the ryanodine receptor. Just whatever they call it, when you read about it, it's the one that's sensitive to calcium. This intracellular calcium that comes in from the calcium surge, that trips open this gate, and you liberate this calcium out to dramatically increase the intracellular calcium levels. Even though it was already inside the cell, it was contained with this membrane structure called the sarcoplasmic reticulum, but you need to get it out. And your author refers to this as the calcium sparks. Okay, just to call it something different because it's a different event. You increase intracellular calcium through the voltage-gated channels, and you increase it more when you open this gate. And that's enough calcium to cause the cross-bridge cycling, and you start to accumulate tension. You'll even reach max tension during this phase. So during the plateau, you start to increase tension, and it even maxes out towards the end of this phase. So just think about the cardiac cycle when you're generating tension in the heart muscle. Is that systole or diastole? Systole? Studied a little bit, you probably figured that out, right? When you generate tension, that's systole. You're squeezing the heart, outflow. So, gotta write this somewhere. I want to write max tension generated. I'm going to erase my picture here. I don't think I need it anymore. But the first event that occurs is this calcium surge right after the sodium channel inactivation that delays the repolarization. So, delayed repol. if you want to put a number to that, if you want to take an average, what would you put it at? 20 to almost, I'd put it at something like 10. You stay depolarized at plus 10. You're not repolarizing to negative potential yet. Because that calcium surge will then cause what I call the calcium sparks. And then max tension is achieved. In that order. The other thing that's happening kind of at the same time, I call the potassium plug. So let's remember, I'm going to draw a couple of potassium channels, green, right? I'll draw a couple of voltage-gated calcium channels right next to it, just to get you to think about this stuff. So they're all embedded in the cell membrane. Okay, remember, calcium, if the gate is open, calcium will enter the cell. If you allow a positive charge into the cell, that contributes to a positive membrane potential with respect to the outside, right? Positive membrane potential with respect to the outside. To reinforce that, remember, potassium, the gradient is set up that if the gates are open, potassium will leave. But you don't want that. You want to maintain this delayed repolarization. So it's like you plug it up. You just plug it up. It can't get out. Potassium tries to get out and it can't. So that way you're keeping positive potassium in the cell as well. And that helps maintain this delayed repolarization. That's all I'm saying. Now, on the bottom graph, where do you see that plug? What I call the plug. I'll let you look at it for 10 seconds and I'll point to it. Sometimes I always wonder if you actually try to do it. It's right there. Now why is that the plug? This is the lowest point of permeability for potassium. It's trying to get out, but it can't. Okay. So you have your max surge of calcium. You plug up the potassium. You're keeping all that positive charge in the cell there. Okay. Well, anyways, everything happens here. All right. So before I move on, is there any questions? Just in your notes, when you go home and write it out, just kind of like get the order. Boom, 1, calcium surge. Boom, 2, I guess 3 and 4. And go to that kind of number. Let's see, am I forgetting anything? I think that's it. The last thing is you allow the cell to repolarize back to rest. Okay, look where I put the number 3 on the bottom graph, and look at the permeability of those 3 ions. What has the highest permeability? Potassium. So now I think you're seeing this the way I want you to. Everything else is closed. You're just letting potassium leave the cell. That repolarizes you. Polarization, it's due to potassium efflux. Potassium permeability is at its highest. It allows you to return to -90. Okay. All right, so this next slide. or I point to number 1, what would you call that if I had you name it, fill in the blank? I would call it depolarization. Depol. What did I call number 2? Yeah, I call it plateau. There's other ways you could state it. You know, delayed repolarization or whatever. 3 is repol. But under 1, 2, and 3, could you place those 6 things that we just talked about? Go ahead and do it now. Just, you know, as a quick review in your notes. Or better yet, it's been a while since we did a half sheet. Let's do this on a half sheet. I'll leave them over here. Actually, let me just, you guys can pass them back. Answer this on your half sheet. Place those 6 events under 1, 2, or 3. All right, so I think it should be done by now. Did you notice that most things, of those 6 things, most of them should fall under number 2?
Yes.
All of them except, well, number 1, the only thing you should have put was the spikes. And for number 3, the only thing you should have put was the potassium efflux. But everything else is under number 2. And there's one thing here that I want you to know that's not listed. So let me point to it. Let me see if you can name what you would call right there. Inactivation of what? Yeah, sodium. I erased it, dang it. But I do want you to know that as well. I just didn't list it on this one. I didn't point exactly to there. So you get your depolarization. It starts to inactivate, then, but it's delayed here. So let's start to get into why you have this delay in repolarization. It's a very unusual action potential as action potentials go. Usually action potentials are more spiky, but this is kind of like has a wide expanse. And okay, let me cut to the chase and tell you why this is important for heart. Now in heart, when the heart's in the condition of rest and it's filling, what do we call that? Starts with a D. Diastole, right? And this allows for that. This allows for diastole. The heart can always have a filling phase. If the heart had no filling phase, it wouldn't pump blood. So this plateau, as I call it, or delayed repolarization. I guess the third way you could put it is prolonged depolarization, or however you want to put it. It allows for diastole. In the heart muscle. Okay, so, well, why? Well, let's keep track of some things here. Let me first start by defining something. It says it right here, refractory period. Refractory period. In physiology, it basically means a period of insensitivity. So in this context, you can't get another action potential during that time. Can't re-stimulate. Can't re-stim cell. Can't get another AP because you got one going on. There's one happening, so within that time, you can't get another one. Okay. You know, in muscle, you could re-stimulate muscle really quick because, well, instead of being 0.3 seconds, in muscle it's like 0.01. It's much shorter. Okay, skeletal muscle, but cardiac muscle, you got this long action potential. This entire time, you're in refractory because of the long plateau. You cannot re-stimulate. So I have a series of graphs to kind of point this out to students. Like for example, this is skeletal muscle, okay? And if you keep track of certain things, when you compare skeletal muscle to cardiac muscle, I would keep track of the action potential duration, how long it is, AP duration. Because that kind of like determines the refractory period. Then think about the muscle fiber itself, the muscle twitch. How long does it take to like build tension and then completely relax? So let's call that contraction duration of the muscle. On this figure, it's that blue line. Contraction duration. And then another thing I'll throw in here is the idea of tetanic contraction, tetany. Is it possible? Is tetany possible? So I'm phrasing it as a yes-no question. But anyways, I would keep track of boom, 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 all these things, and kind of noted each for skeletal and for cardiac muscle. Well, what you could do is let's just kind of take it point by point here. Let's see where I'm going with this. So basically, what you do in the lab is you take your muscle, your muscle fiber, whatever it is you're testing, and you stimulate it. You just zap it with some voltage. Okay, and, we don't do this wet lab. I wish we did. I did it as a grad student. Students would kind of like dissect out. I'd have to kill the frogs and then kind of dissect out. Kill sounds so cruel. Well, we say sacrifice. But I put their head in a guillotine and chop it off and pith. That's what we did. Okay, and the students would do the dissection. Now we just have computer simulations, and we don't have to do that anymore. But what students did was they would like put these electrodes in the muscle and zap it, or sometimes they put electrodes on the nerve and stimulate the nerve to make the muscle twitch. And in that experiment, what you would do is you would see that you get the red action potential. The action potential is very quick. It's like 10 milliseconds here. And that basically determines the length of the refractory period. It's about 10 milliseconds. But the contraction duration is much longer. It takes about 100 milliseconds to completely build tension and then completely relax. And you should know the answer to this if you remember your skeletal muscle physiology. It's a big yes. Skeletal muscle is supposed to have this nice tetanic contraction where if in the lab, if you re-stimulate muscle, you want to see how much tension it can generate. And so if you stimulate, you set your amplifier to kind of re-stimulate every, 75 milliseconds. What you're doing is, at 75 milliseconds, at 75, you're stimulating first here at 0, and as it starts to relax, you re-stimulate again before it has a chance to get to 0. And as a physiologist, you're asking the question, what's it going to do? There's still tension in the muscle, and if you zap it again, because you're outside the refractory period, what we wanted you to see in 430 is that you get this kind of hump thing, and we call that the wave summation. Eventually, you'll build enough tension, you get this nice smooth line, and this is how muscle operates. This nice smooth maximum tension. Okay, so that's tetanic contraction in the skeletal muscle. Any questions on how that worked, that laboratory experiment? Because now we're going to look at cardiac, and we'll see it's different. All right, this one here, same graph, but I answered the last question here is not possible. So let's write that answer in and kind of build up to it. No exclamation point, that's a good thing. That's a good no. No is good. All right, so let's kind of look at this. As we study the action potential, we said that there's a very long refractory period because the action potential duration from boom to boom, it's about 250 milliseconds. It's very long. That's why I wanted to kind of put this data side by side so you can see that 250 milliseconds is much longer than 10. And so the refractory period is about as long. About, some books will say 200, or it doesn't matter. It's much longer. Okay. 250 is much longer than 10. Okay, that's all I'm trying to say. Now, the thing is, the contraction duration is about as long. You build tension, and then you completely relax. Maybe a little more than 250. I'll just say 250 plus. It's a little more. But the point is, if you can kind of see where I'm going with this, you're just writing numbers down, but are you processing what's going on here? When the refractory period is almost as long as the contraction duration, that tension is going to be completely gone in the muscle. So that when you do the experiment where you try to make it do the tetany thing, You can't do it. You stimulate at time 0, and do you see where they gray out the stimulus things there? It's like you're zapping it to re-stimulate, but you can't do it because you're within refractory period. So when's the first time you can re-stimulate? At about 250. Okay, and then do you build tension? No. You just kind of stay the same. So what they're trying to tell you is tetany is not possible. Let me turn the mic down a little bit. Okay. All right, so I mean, all that having been said, could you answer this question? How does a longer absolute refractory period prevent tetany? Well, what I'm saying, let me write out my logic. I'm going to erase my data slide here, my data table. So the train of thought I'm trying to get you to go with is, we already got it here. You got this refractory period. So you have a much longer AP. Therefore, you have a much longer refractory period. You can't re-stimulate. You can't get another AP. You can't re-stim. So then the logic goes, okay, well then the muscle, when it contracts, it has time to completely relax. This is the diastole, okay? So this allows for inflow when the muscle has time to relax. Okay? I mean, what is diastole? The muscle's relaxed. You can feel again, right? Ensures there will always be a filling phase. It's all because of those voltage-gated calcium channels making the active potential longer. So it starts with this. That's kind of the train of thought there. It's really, well, if you think about it, what if the heart, if you achieve tetany and you get this tension, you just stayed in systole, okay, you pump blood once, but you're in systole. Can you refill when you're in systole? There's too much pressure in there. You're not going to feel it, okay? So that'd be equivalent to, well, okay, there's something called ventricular fibrillation. It will be like that. Ventricular fibrillation is when the electrical signal doesn't spread correctly. And I've been told if you could have that heart in your hand and you felt defib, it feels like a bag of writhing worms. You get no cardiac output. You probably get no inflow. Okay, that's why death is imminent if you don't correct that. And that's when they put the defibrillators across. They shock it. What you're trying to do is you're trying to short circuit the heart. And you hope the SA node kicks in after you shock them. Okay, you've all seen it, and these on TV shows, and you put the paddles on, and they say clear. Now, why do you think they say clear? If you're touching them, you're going to get that shock too. You don't need it because you're alive, So that's why this is important if it's working correctly. You always have a diastole. Well, I spelled that wrong. Diastole, sorry. let's move on here. Like, for example, on your half sheet, what's wrong with this thing? Let's call this, after your little table there, just call it number 1. You're just choosing A or B, and I'm asking you to look at it and say which one's the cardiac muscle. Just give it a once over and say, oh, okay, that's probably it. I categorize this question as easy, if you're paying attention. All right, so you answered it, what'd you put? B is correct. You got the easy one right. Now it gets a little more nuanced here. Let's call this number 2. Answer it on your half sheet. I'll let you look at it. We're choosing from A, B, C, D here. The absolute refractory period for cardiac skeletal muscle. I'm not going to give you the correct answers here. I just want to see if you can get it when I grade your half sheet later. All right, so for this one, if you got that previous one right, don't answer this on your half sheet. But what would you put for this one? Which has a longer absolute refractory period? Yeah, cardiac is correct. So if you know that now, you probably answered this one correct. So I'll check it out later. Now that's what I'm going for here. They kind of put this little green shade to illustrate refractory period is much longer here than up there. That's the whole point of this part of the lecture, to understand that. And but I do want to shift gears from contractile cells to autorhythmic cells. I'll do a deep dive on that. Let's see. Okay, so how are we doing? Good. So let's switch our minds here. Now we're going to talk about action potentials of these cells here, primarily in the SA or the AB node. The Purkinje fibers, they do have some pacemaker potential too, but we're not talking about those for the action potential trace we'll be studying. So the idea of pacemaker activity is that there's this, they call it a spontaneous depolarization of the cell membrane that leads to an action potential. Any cardiac cell with pacemaker activity can initiate the heartbeat. And the one you want to operate, the strongest one, SA node. That's the sino-atrial, the normal sinus rhythm that establishes the heartbeat. So what they do here is they repeat themselves. They want students to recognize the different potentials, what carries the current, which we already talked about, and then which gates carry the current. Okay. So as a review, what's the bottom number they start at? You probably can't read it in the back. They start way down there. They start -60 on this graph. And they go to -40 in terms of millivolts. They're going to call that their threshold potential. I'll just draw a dotted line here. That's enough depolarization to get an action potential. So what we had said prior is that we know that that pacemaker potential is carried primarily by sodium currents. Right there. I used yellow. I'll call it number 1. Number 1, we call it a pacemaker potential. It's a slow rise to threshold. carried by sodium current. You know, sometimes some physiologists have called that current a funny current carried by funny channels. They say net sodium in. They say some calcium's let in too, but it's primarily sodium. And when they first observed these, they thought it was funny how they kept measuring these currents. So they call them funny currents, and the channels that carry them, funny channels. I think it's the only time I've seen the word funny make it into the textbook. It's carried literally by sodium, but they're called funny currents sometimes, and you may see the term funny channel. Now, they're also called Hcn channels. So I would know all of that. Okay. The H stands for hyperpolarization. The Cn is like something activated, cyclic nucleotide gated, some other stuff. Don't worry about it. But I think you should understand that. Hyperpolarization, that's when these channels open. Whenever the cell becomes hyperpolarized to like -60, you can't stay there at rest. They open and you have a slow depolarization to threshold, -40. Okay, so that's the key there. That yellow line keeps your heart beating. Okay, it's really important. All right, so let's talk about the action potential. And there's two parts. The action potential is you depolarize and repolarize. But we now know that the depolarization is carried by calcium. Okay. Let's call it number 2. Calcium is responsible for depolarizing the cell. And then you repolarize, so there's no delay. That's caused by a potassium efflux. So calcium is responsible for the repolarization of the membrane potential. Anyways, the dipole, repole, that's our action potential. Okay, straight up. Dipole, repole. Boom, that's it. Okay, and as soon as you get back to a hyperpolarized state, those funny channels, the Hcn channels, they open up and you have that slow rise to threshold again. Okay, you just go around. I won't draw it again, but you get the idea. You just keep depolarizing, then slowly depolarizing the threshold and get your action potential. Okay. All right. But we covered all that, I think. So that's kind of how, this is the figure from Merriam, and they go through pacemaker, dipole, repole. I just wanted to give you that figure because it's straight from the book. Let's do another half-sheet table. Call this one table number 3. It'll be a good review to compare those two action potential traces, because there are some differences. So I have some things written up there. I have pacemaker potential. pacemaker POT, dipole, plateau. I have repolarization and tension. So repol, tension. And I just threw these up there as terms to talk about contractile versus autorhythmic cells of the SA node. Starting with pacemaker potential, let's just, which cell had it? Only one of them had it. Boom, autorhythmic cells had it. I never discussed the pacemaker potential in the contractile cells. Does not apply. Not applicable. Now, both cells had a depolarization. However, the difference is carried by different ions. Do we remember what carried the depolarization for the autorhythmic cells? I'll let you fill it in for that. Okay. Plateau. Which action potential had it? Contractile. And what was the primary reason? Primarily because of calcium. Okay, autorhythmic cells not applicable. It didn't have one. Both cells, of course, repolarize, but what ion are you going to put? I'll let you fill it in. And of these two cells, which one could generate tension? Just mark which one. It's only one of them. I'll see if you get it right. Next topic, more with autorhythmic cells. You know, you can vary your heart rate. You were supposed to see that in the lab last time. When you were lying down and your subject sat up, you were supposed to see an increase in heart rate. And I'll have you turn in your lab later today. I will look for that from segment 1 to segment 2, because you create a transient drop in blood pressure. You get that lightheaded feeling, and the heart starts beating faster to compensate. The point is that you have an accelerator and the brakes. The sympathetic and parasympathetic, they can modify your pacemaker. This is the physiology I want you to understand. Actually, let's tackle this after the break. Come back in 15 minutes. And we'll continue lecture.